Today, we'll be doing a mind map on DNA. DNA is short for deoxyribonucleic acid, which is a molecule found in almost all animal cells' nuclei which carries and stores genetic information. DNA is made up of basic units called nucleotides, which are made up of three main components: deoxyribose sugar, phosphate group, and a nitrogen-containing base. The base structure of a nucleotide in DNA is like this. If you were to uncall this double helix structure of DNA, the nucleotide sequence will look like this. DNA is made up of two anti-parallel polynucleotide chains. Nucleotides are connected to adjacent nucleotides on the same strand via phosphodiester bond, and this bond occurs between the 3' and 5' ends of the nucleotide. When a free nucleotide is added to the strand, it attaches its 5' end to the free 3' end and forms the phosphodiester bond. A free nucleotide, also called deoxyribose triphosphate, has three phosphate groups attached to it. However, when a phosphodiester bond forms, the bond with two of the phosphate groups will be broken. The application of this will be seen when we go into DNA replication later on. Additionally, as you can see, DNA has four different nitrogen-containing bases: adenine, cytosine, guanine, and thymine. The sequence of the anti-parallel chains follow the rules of complementary base pairing, where adenine is always paired with thymine and cytosine always with guanine. The complementary bases are held together by weak hydrogen bonds, where between adenine and thymine there are 2, and between cytosine and guanine there are 3. When the nucleotide bases are bonded as such, DNA naturally forms the double helical shape we are all familiar with, due to the anti-parallel nature of the DNA strands. Along a strand of DNA, there are multiple genes, which are specific sequences of DNA nucleotides of varying lengths. Each gene or sequence controls the formation of a single polypeptide or protein based on the genetic code stored within the gene. As a gene is a specified length of DNA, it consists of two polynucleotide chains: the template strand, also known as the antisense strand, and the coding strand, which is also known as the sense strand. The template strand determines the protein synthesized, as its nucleoside base sequence determines the amino acid order which makes up the polypeptide. A sequence of 3 bases is called a codon, and it corresponds to a specific amino acid. This process of gene expression will be explained later on when I discuss transcription and translation. Now to DNA replication. The structure of DNA was discovered by Watson and Crick in the 1950s after seeing an X-ray picture of DNA produced by Rosalind Franklin through a technique called X-ray crystallography. There were initially 3 theories on how DNA replicated: conservative, semi-conservative, and dispersive. However, an experiment by Messelson and Stahl soon proved the semi-conservative method of replication as the correct method of DNA replication. This is something you can read up more on, as it may come out as a Nature of Science question in your exams. How DNA replication begins is enzyme DNA B locates and marks out the initiation site on the DNA molecule where replication begins. Once done, DNA gyrase attaches and relaxes the supercoiling of DNA. DNA helicase then unzips the DNA strand by breaking the hydrogen bonds between complementary base pairs. This creates a replication fork, which separates DNA into the leading and lagging strand. Single-strand binding proteins hold the separated anti-parallel strands apart to allow new nucleotide chains to form. Let's first talk about the replication of the leading strand. DNA polymerase III is the enzyme that replicates DNA. However, it can only add nucleotides to an already existing strand and cannot initiate its own chain. This is something important to note. What happens then is that as DNA is being unzipped, RNA primase constructs and attaches RNA primers complementary to the start of the leading strand. An RNA primer is simply a sequence of 10 to 15 nucleotides that DNA polymerase III can add nucleotides to. Therefore, once the RNA primer is in place, DNA polymerase III attaches itself to it and begins adding nucleotides in a 5' to 3' direction, complementary to the base sequence of the leading strand. the direction of replication is towards that of the replication fork, and this replication occurs until termination. The direction of replication is always from a 5' to 3' direction. On the leading strand, this means that DNA polymerase III adds nucleotides towards the direction of the fork. However, on the lagging strand, due to the anti-parallel nature of DNA, the direction of replication is away from the fork. Hence, the mechanisms for forming the nucleotide chain is different. As DNA helicase unwinds and unzips the DNA molecule, RNA primase, which we already touched on earlier, leaves many RNA primers along the lagging strand. DNA polymerase III attaches itself to these primers and adds nucleotides in a 5' to 3' direction, which is away from the fork. This continues until it reaches a previously placed primer. When it does, it detaches from the nucleotide chain and is replaced by DNA polymerase I. This enzyme then removes the RNA primer and replaces it with DNA complementary to the base sequence on the lagging strand. This forms multiple fragmented nucleotide chains along the lagging strands called Okazaki fragments. To combine these fragments to form a continuous nucleotide chain, DNA ligase attaches itself between Okazaki fragments and forms phosphodiester bonds between them to form a continuous replicated DNA strand. Due to the rules of complementary base pairing, both DNA strands are now identical to each other and identical to the original DNA molecule that was replicated. This is how DNA replication occurs. Looking at the diagram, I drew DNA polymerase III as two separate enzymes replicating DNA along the leading and lagging strands separately. I only did this to illustrate the process. In reality, DNA polymerase III is not two separate subunits. Instead, what happens is that during DNA replication, the lagging strand loops itself so that DNA polymerase III is able to replicate both the leading and lagging strand at the same time. This is just an interesting point to note. Now, we move on to transcription and translation. As discussed earlier, a gene controls for the formation of a specific polypeptide, and gene expression is the process in which a polypeptide is formed. This process is split into transcription and translation. However, before we go into the mechanisms of these two processes, we have to understand the order of the operon, which controls gene expression. An operon is a functional sequence of DNA containing a cluster of genes under the control of a single promoter. What this means is that multiple genes for different polypeptides may be controlled by the same promoter. The operon has three main regions: the promoter region, the operator region, and the gene region. The operator region is commonly associated with a repressor protein. When a gene is to be expressed, RNA polymerase attaches itself to the promoter region first, which is usually identified by a TATA base sequence. However, whether transcription of the gene can occur is decided by the operator region. In the presence of the repressor, RNA polymerase cannot move onto the gene region and begin transcription. Only in the absence of the repressor will RNA polymerase be able to move through the operator region to reach the gene portion. This is a little confusing, so let me give you an example. The gene for production of lactase enzyme is expressed only in the presence of lactose, because lactose attaches to the repressor protein and causes it to break off, thus allowing RNA polymerase to move into the gene portion and begin transcription. Okay, so now to transcription. RNA polymerase is signaled to attach to the promoter region of an operon by a transcription initiation factor, also called a sigma factor. Once attached to the DNA strand, the enzyme begins unwinding the double helical structure of DNA. This separates the coding and template strands. Transcription begins complementary to the template strand, where three nucleotides are joined together to form a messenger RNA strand. The same rules of complementary base pairings are followed as in DNA replication, except that RNA does not contain thymine. Instead, it contains uracil, where uracil now pairs with adenine. So, for example, if the sequence of the template strand is AAGCTC, The mRNA base sequence complementary to this will be UUCGAG. RNA polymerase moves along the gene until it reaches a rho recognition site, where a rho protein attaches itself to the mRNA strand and it begins moving along mRNA toward the RNA polymerase enzyme. Once RNA polymerase reaches the termination site, the rho protein will reach and bind it to RNA polymerase. This causes the formed mRNA strand to detach from the enzyme. Before this mRNA molecule leaves the nucleus to begin translation, it may undergo splicing. This process is carried out by spliceosomes which remove non-coding regions on the mRNA strand. After removing the introns, what is left is referred to as mature mRNA, which then leaves the nucleus via the nuclear pores. The next step of translation occurs at ribosomes in the cytoplasm. Once in cytoplasm, initiation of translation occurs when the mRNA strand binds to the smaller 40S ribosomal subunit. The mRNA attaches itself at its start codon via means of complementary base pairing. The common start codon you will come across has the sequence AUG. Polypeptides are made up of amino acids which are brought over to the ribosomes by tRNA molecules. This is a simplified form of a tRNA molecule. The initiator tRNA, whose anticodon is complementary to that of the start codon, binds to it. As this occurs, the largest 60S ribosomal subunit attaches to the mRNA molecule such that the initiator tRNA is in the P site. The ribosome has 3 sites in total: the amino acyl site, referred to as the A site, the peptidosite, referred to as the P site, and the exit site, referred to as the E site. Once the 60S subunit attaches itself, elongation of the amino acid chain begins, where a tRNA molecule whose anticodon is complementary to the next mRNA codon enters the A site. A peptide bond begins to form between the amino acids attached to each tRNA molecule. Once this peptide bond forms, the amino acid attached to the tRNA molecule in the P site breaks off. The ribosome then moves down the mRNA strand, where the deacylated tRNA is now located in the E site. Once in the E site, the tRNA is released, where it can attach to an amino acid again. The next tRNA molecule moves into the now empty A site and the process repeats, elongating the polypeptide. This continues until the ribosome reaches the stop codon on the mRNA, which is either a UAG, UAA, or UGA sequence. One important thing to note here is that the ribosome is the one that moves down the mRNA strand, and it is not the mRNA strand that moves. This is the same for transcription and DNA replication, where the respective enzymes are the one that moves down the DNA strand as it creates the nucleotide chains. Once the ribosome reaches the stop codon, a release factor enters the A site and terminates elongation of the polypeptide chain. The polypeptide chain breaks off from the ribosome and the ribosome detaches from the mRNA molecule. At this point, translation is completed and the polypeptide is formed. This brings me to the end of my mind map on DNA. I hope you find it useful and have a good day.